Hi, y'all. It's Dr. Bree. Today I'm going to be taking you through a whirlwind tour of the forces of evolution as part of our series on the introduction to biological anthropology. So we'll start with talking about population genetics. We have already covered in previous lectures that evolution is not a population level thing. Although we measure evolution via allele frequencies, that is an aggregated thing, right? I can't measure the change in allele frequencies within a singular person. I have to measure them within a groups. Thus, evolution has to take place at the level of populations. Evolution doesn't occur within a singular person or even a singular lineage, really. It takes multiple individuals in a breeding population to work. So remember that a population is a group of individuals of the same species that are geographically close enough to one another to make it viable that they can interbreed with one another and produce fertile offspring. Recall that if they do not produce fertile offspring, that does not qualify as a species. So remember the liger and the mule, those are both hybrids, meaning that those two species are close enough to actually have an offspring because their genetics are that similar that it would not be fertile. It might be viable, it might live to adulthood, but it is not fertile and cannot have another offspring. So we discuss populations with regard to a very small unit, though, the gene. So it's interesting that we have this macro level perspective along with this micro level perspective at the same time. Now, genes, although we've been talking about them through Punnett squares and pedigree charts, I think it's important to give you a discrete definition at a couple points throughout this class. And so this is one of those points. A gene is defined as the basic unit of information that encodes the proteins needed to grow and function as a living organism. So proteins are essential for the genes, basically. So our bodies code for genes, genes create proteins, proteins create what we see on the phenotype on the outside. So here on the screen, I've got a number of levels where you can see the micro level structure, the DNA, that becomes, if you have enough DNA that codes for enough amino acids and protein, and thus proteins, you get a gene. A gene is located on the chromosome, and these chromosomes are located within the nucleus of the cell because we are eukaryotes, not prokaryotes, so we have a nucleus. So each gene, as we have learned through Punnett squares, can have multiple alleles. These alleles are just variants of that gene, each of which can produce a slightly different type of protein, which again manifests in a different phenotypic variation. So each individual carries a collection of genes that can be passed down to future generations. You will not get all of your parents' genes, you will get a selection, which is why I use the word can. So we can think of populations then as gene pools, like big, yeah, I mean, you can imagine them as a pool if that is helpful, a physical pool. And inside of that physical pool that is in your mind that you're visualizing are all of the possible alleles that offspring can inherit in a breeding population. So, the scientific definition of a gene pool is the entire collection of genetic material in a breeding community that can possibly be passed on down to the next generation. So, on my screen, we are looking at a gene pool. It's a circle with a bunch of X's in it that are various colors, and that's just representing the different forms of a gene that exist within this breeding population. And below that, we've got three different circles that show us Gen 1, Gen 10, and Gen 20. And just by looking at the colors, even if we don't look at the letters within them, we can see that the allele frequencies are changing over time from generation 1 to 20. We've got no red alleles in generation 20. We've got no red alleles. But we started with about half and half in generation one. So that means from generation one to 20, there must have been some sort of force of evolution working that the red allele, the dominant allele here, because it's shown with capital letter A, so we know it's the dominant allele, that there must have been something going on with that allele to make it disfavored in this population. So this is a visual way to understand how allele frequencies changing over time represent evolutionary trajectories. So a reminder, you should remember this, but as a reminder, evolution is defined currently in the modern era as change in allele frequencies over time. So therefore, if allele frequencies were staying the same from one generation to the next, the population would not be evolving. So allele frequencies, we calculated genotype frequencies in the last lecture for Punnett squares. So like 1 out of 4 offspring, that would be 25% probability, that kind of thing. Now we're going to be looking a little bit more specific at the alleles themselves that would make up that genotype, and we're going to be calculating allele frequencies. There is not a whole lot of math today, do not worry. So the allele frequency is the ratio or percentage of one allele compared to the other alleles for that gene within an entire population. Now, if you are like me, you're probably like, that makes absolutely no sense. Don't worry, I'm required to give you the scientific definitions, but I'm going to show you what those mean because like you, I'd just be like, and not actually understand what that meaning is. So we're going to work through a problem so I can show you what this actually looks like and how to calculate it. In this particular problem, we are looking at different flower colors. We have got, in this one, we have got incomplete dominance going on. How do I know that? Because the heterozygous condition, oh, let me whip out my pen. All right, the heterozygous condition here, the pink flower, is a blend of the dominant condition, which is red, and the recessive condition, which is white. And in the last lecture, we learned that if the heterozygous condition is a blend, that's actually incomplete dominance, that it means that the capital R, the dominant allele, cannot fully dominate the recessive allele, which is in this case the white allele that codes for a white flower. So that's not really relevant, but I do like to make connections between my lectures to kind of help reinforce the concepts. All right, so we're looking at flower color, right? So our possibilities are red, pink, and white, and we're going to be calculating allele frequencies within this population. So we have got a square on the screen. Within that square is going to be our gene pool. Inside of that, I guess it's a rectangle. Inside of that rectangle, we have got 10 total flowers. Now, for Mendelian traits, remember you always have 2 alleles, one from mom, one from dad. So for every flower in this population, they have 2 alleles. And you can see that visually underneath them. So for instance, this red flower has capital R underneath it. That represents the 2 alleles. So if we were to add up the total number of alleles inside of this rectangle, inside of this box, how many would there be? Now, there's 2 ways you can do this. Number 1, you can just count the number of letters that you see in this box. Number 2, you can work it out quickly mathematically, knowing that there are 10 individuals with 2 alleles each. And if you do that multiplication, there are 20 total alleles in this entire population. Okay, so down here in this, I have got a red box that says allele frequencies. We're going to calculate the frequency of capital R, the dominant allele, and the frequencies of lowercase r, the recessive allele. So we are going to use for a denominator in these ratios, we are going to use 20 because 20 is our entire amount of alleles in this entire box. That's our entire gene pool. So the first thing we're going to do is figure out how many capital R's or dominant alleles we have in this breeding population. So you can just go through and you can count. So I've got 2 big R's here, 1 big R here in the heterozygous. So 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I've got 12 capital R's. Okay. And I'm going to divide that by the 20 possible alleles in this entire population. We do that math. I can't do that in my head, but if you plug that in a calculator, you're going to get 0.6. I just looked right here, just in case you're wondering. I'm not great at mental math. I'm the kind of person that puts like 2 times 2 in my calculator just to make sure it's still 4, just to make sure nothing changed. So I'm smart in some ways. I am not so smart in other ways. So this is also 60%. These are the same thing. All right, so now we're going to calculate the allele frequencies of the recessive allele, the lowercase r, and I already know what it should be because these 2 numbers, again, need to add to 100%. However, I'm just going to double check my math to make sure that's actually the outcome that we get because this is exactly what I would do if I were taking a test. So we count the number of small r's that we see, the lowercase r's. We've got 1, 2, 3, 4, 5, 6, 7, 8. So I've got 8, and 8 recessives plus 12 dominants does give us the total of 20 that we are looking for. If those two numbers, if this was like 13 and 8, that gives us 21. That can't be possible because there are not 21 alleles inside of this rectangle. So go back and count them again. Now, the harder types of problems that I would give with these is I would describe the conditions to you. So I would say there are 20 homozygous dominant individuals. And so instead of you already seeing that there are two capital R's for each of those individuals, you would have to calculate that yourself. So I like to throw in some word problems that are not super complex, but just to make sure that you know that if this question says homozygous dominant, that those individuals have two capital R's or two dominant alleles. Anyway, coming back to this recessive allele, 8 out of 20 is going to be 0.4. Again, I did not do that in my head, and that should be 40%. 60% plus 40% gives us 100%. So we are pretty sure that this is correct, and I will validate you if that is correct. All right, now we're going to work through something else that shows generations so that I can show you what this looks like over time. So we are going to determine the allele frequencies of the dominant and recessive alleles for each generation over time, and we're going to figure out how much they change over time. So here are our three different generations. In this instance, you don't have 10 individuals. This is a much smaller breeding population. So our denominator is going to be different. And that's where I like to start each time is figuring out what my denominator is. And in this particular example, you've got the same number of individuals in each generation, which is fairly likely in real life, but let's not think about that. So our denominator will stay the same across time. But so we've got 5 individuals in each of these circles. So that means total, we've got 10 alleles. Each individual has 2 alleles. 5 times 2 is 10. You can also just count the number of letters that are in the circle and you will still get at 10. So 10 is going to be our denominator, and that's super awesome for us because dividing by 10 is pretty easy. Okay, so now step 2. We've already figured out that there are going to be a total of 10 alleles overall in this gene pool and for all the rest of the gene pools. Now we're going to count the number of dominant alleles that we have here, and then later we will count the number of recessives. So the number of capital R's, how many do we have? And I'm gonna phrase this how I would in a word problem. I would say there are 3 individuals that are homozygous dominant, 1 heterozygous individual, and 1 homozygous recessive individual. And from those verbal descriptions, you would need to figure out that this is actually what we're looking at. We've got 3 individuals with 2 capital R's, so that gives me 6 dominant alleles. And then I get 1 more dominant from this heterozygous individual, so that gives us 7 total capital R's. In this problem on the screen, you can simply count them. I'm just telling you what the next step up would be to test your understanding is to describe these without giving you the actual image. I would describe them in words. Okay, and then I know that there are 10 alleles total. 2, 4, 6, 8, 10, right? Counting them. And that's math is really easy, and that is math that I can do in my head. That is 0.7, because dividing by 10 just tells us to move the decimal from behind the 7 to the front of the 7, or 70%, right? Or 70%. So we already know what our allele frequency should be for the recessive alleles, right? So if it's got to add to 100, I'm expecting 30%. However, gonna check my work. I've got 1, 2, 3. I've got 3 small r's or 3 recessive alleles, and that does in fact give me 0.3 or 30%. So those are our allele frequencies for Gen 1. Now let's hop on over to Gen 2. I will change my color so it's easier to tell them apart. All right, so we still have 5 individuals, so we've got 10 total alleles in this gene pool. So now we're going to, again, just count up the number of capital R's we have and the number of recessive. And I will tell you how I would phrase this as a word problem. I would say there's 1 homozygous dominant individual, 3 heterozygous individuals, and 1 homozygous recessive individual. What are the allele frequencies of the dominant and recessive allele? So the homozygous dominant individual gives us 2 capital R's. I've got 3 heterozygous, and for each of my heterozygotes, they're going to give me 1 dominant allele. So that's going to put me up to 5 total. You can count these as I'm underlining them on the screen. So 3, 4, 5. That's it. I've got 5. We've still got 10 total alleles. So as a percentage, that is going to be 50%. Okay, so we're already expecting the recessive allele to be what frequency? 50 plus what gives us 100%? 50, but we're just going to double check it because we are nervous about getting this wrong on our test. So we've got the homozygous recessive individual gives me 2 little r's or 2 recessive alleles. And then each of my 3 heterozygous individuals gives me 1 more recessive allele. So that does give us a total of 5. So you can count those, you can pause and count those if you want, or 50%. Okay, so look, let's just see how much we've changed from Gen 1 to Gen 2. Let me yet again change my color of my pen. So our recessive allele, and I'm going to put delta here. Delta just means change in, and it's used in physics. It's just a shorthand for that. It just means the change in. So delta is a little triangle. It's symbol for change. Okay, so the change in the dominant allele frequencies from generation 1 to generation 2, we went from 70% down to 50%. So that's a decrease or an increase? A decrease. So we went from 70 to 50. That's a decrease. So we know it's going to be negative. I'm just going to put this as a decimal just to save myself space, but it would be the same as a percent. So we decreased by how much? From 0.7 to 0.5 is 0.2. So we decreased 0.2 or we decreased by 20%. So what does that mean for the recessive allele? What do we think that means for the frequency of the recessive allele? How did that change? Remember, all the time it's got to add to 100. So what needs to happen is that the recessive allele needs to increase by the same amount that the dominant allele decreased. So if my dominant allele decreased by 20%, to keep all of my numbers adding to 100 for each gen, my recessive allele needs to increase by 20%. And we can see that my 0.3 has gone up to 0.5, or my 30% has gone up to 50%. So that went up by 0.2. All right, heading now to Gen 3, we're going to figure out the allele frequencies there. Let me change my pen. So we've got 5 alleles, which means we've got 10 alleles overall in our gene pool. Again, we're going to count up, and I don't have quite so much space over here. How many capital R's do we have? Our dominant alleles. We've got 2, 3, 4. We've got 4 of them. To phrase how I would say this, I've got 1 homozygous dominant individual who gives me 2 dominant alleles, and I've got 2 heterozygous individuals that each give me 1 dominant allele. Add those together, you've got 4. Added 10 gives me 0.4 or 40%. 0.4 or 40%. So already we are thinking about what our allele frequency for our recessive allele should be, and what should that be? 60%, because 40 plus 60 equals 100. So we'll just double check. We've got 2 homozygous recessive individuals. Each of those individuals gives us 2 recessive alleles. So 2 plus 2 gives us 4. And then we've got 2 heterozygous individuals, each of which give us 1 recessive allele. So that gives us a total of 6. 6 out of 10. And you don't have to do that in your head. You can totally write out each of the genotypes and then just count up the letters if you prefer that. I'm all for that. Whatever works for you. So 6 out of 10 for our recessive alleles, which is 0.6 or 60%. Running out of space, we're going into the corner here. Okay. So then between generation 2 and generation 3, how are our alleles changing? I'm going to change this back to purple. Okay, so between Gen 2 and Gen 3, our dominant allele has changed from 50% to 40%. So now we've decreased another 0.1 or 10%. That means our recessive allele must have all increased. So if one of them decreases, the other one needs to increase, and the amounts are going to be the same. So we expect that our recessive allele should increase about 10% or exactly 10%. And that is what we see. So 50% to 60%. And oftentimes I'll probably ask you, like, between Gen 1 and Gen 3, what does that look like? But that's the extensive math we're going to do today. So you made it. Hopefully that makes sense. I'm a visual person, so seeing somebody work through the problems is helpful to me. All right, now we're going to hit the four forces of evolution. Those are mutation, which you should already be somewhat familiar with, genetic drift, gene flow, and natural selection. And taken together, all of these four guys count for all genotypic variation that you see in the world today. So we'll start with mutation since this is the one that we have covered already. This is the original source of all genetic variation and the only source of new variation. You cannot get a new trait or a new version of a gene without mutation occurring. Remember that mutation is some sort of copying error in the genetic code, which gives us a new version of a gene, which is called an allele. In a previous video, I went over types of mutations. I will kind of reiterate some of those though. So when most people hear mutation, they tend to think it's bad, inherently bad, like cancers or heritable diseases, genetic diseases. And these are called deleterious mutations, meaning they produce negative effects. And that is true. Mutations can have negative effects. But mutations can also be beneficial or advantageous as well. For example, the mutation that allows Chihuahuas and other tropical adapted dogs that live in like hotter climates, to have thinner coats than you see in wolves and huskies, dogs that tend to live in colder climates or were adapted to live in colder climates, that was a mutation and it allowed them to live and survive in different areas of the world that they didn't before. Remember that all dogs, even if you have a Chihuahua, are descendants of ancestral wolves. So there must have been some change in the coat at some point, which allowed them to not overheat in super hot conditions like Southern California, where I live. I guess it's not as hot here as it is other places, but. So every single person has mutations in their body. Most of them are neutral, so we don't even notice them, but some of them we do notice. Mutations can come about a couple of ways. They can happen spontaneously, like randomly in your body. They can also be inherited from your parents. They can also be generated at birth, just randomly as well. UV radiation and skin mutations do have a known connection. What happens with UV radiation is that it basically causes these weak bonds that are in your DNA that I covered in the like transcription translation part of the last lecture. But it causes the bonds to do something to like separate and then to rebond in a weird way that they shouldn't be doing. And this is called cross-linking. So basically, they should be pairing with the base across from them on the opposite strand, but the UV radiation causes those the bonds to separate, and they bond the pair like on their own strand that's kind of next to them. And this is what you're seeing on the screen is that UV radiation causes this to happen, and this is what it looks like after. And this is called UV cross-linking, and it is not awesome, right? You imagine that your DNA shouldn't really be doing this. So this is what eventually can cause skin cancer to occur. However, many mutations are detected and corrected by DNA repair mechanisms. They feel these things happen and then they're like, nope, I'm going to put you back in line, buddy, where you go. These are little enzymes that patrol and repair the DNA in living cells. And sometimes this repair causes some UV radiation to manifest as like a freckle or a mole. Sometimes there are too many changes. Like if you use tanning booths a lot, like your body cannot keep up with the amount of cross-linking that's occurring. And so that's when we start to head towards the cancer route. Like your body its regulation mechanisms cannot keep up with the changes that are happening in your body. So most of the mutations that occur within you occur within our species, occur within organisms in general, occur in our somatic cells. I'm hoping you remember what somatic cells are, but in case you don't, a reminder is that those are your body cells. So there are simply more of your body cells than there are your sex cells or your gametes. So based on probability, most of these mutations happen in your body cells. And that's good because like things that are bad that happen in your body cells, like skin cancer, right? You can't pass that along to your offspring. So the other main cell type in the body I just mentioned, but those are gametes, those are your sex cells. So only mutations that occur in those sex cells are passed on to the next generation. So that mutation has to be present in specifically the gametes to be able to be handed down, to be a heritable mutation. So when a new mutation pops up randomly in a family lineage, so your parents didn't have it, but you developed it, that's called a spontaneous mutation. And if an individual is born with a mutation and passes it on to their offspring, that means it must have been in their gametes already, and that is an inherited mutation. So I want to give you a visual sense of what this looks like. So I'd like you to take a second to look at this image. And I've circled where the mutation occurs, but I want you to tell me, is this inherited or is this spontaneous? Did this individual on the screen, did they inherit this from a parent or did it spontaneously occur in their body? I'm going to give you a second to think or pause and think. So what we see in the image is we've got parental gametes. Do the parental gametes, the sex cells of the parents, the sperm and the egg, do they have this mutation? No. So what happened is that the sperm fertilized the egg, and then while this embryo was developing, the little you was developing, a spontaneous mutation occurred. So it was not inherited, it occurred during development, but because wasn't carried in either the sperm or the egg, that's not where you got it from. It happened in you as a fetus. That is a spontaneous mutation, not inherited. So this is somatic though. It's happening in the somatic cell. So you can see the correlate where it was in the embryo to versus you as a grown human being. It's in a part of the body, meaning that it doesn't get passed on to the offspring. So you can see a couple of the things that I'm talking about in this singular image. So there are a few different kinds of mutations. I went over like the general umbrella terms for them, like insertion, deletion, substitution, that kind of thing last time. But there are a little bit more specific terms that are used to describe mutations. For instance, if there's a singular nucleotide change, so like one of the bases is swapped, for instance, those are called point mutations, meaning they happen at one specific location in the genetic code. These changes can be major, minor, they can make no difference. Minor changes occur when things like polarity stay the same, but perhaps a different amino acid is coded for, but it's got the same kind of chemical properties as the previous one prior to the mutation. That would be like a minor change. Point mutations that alter the overall protein that are produced are called non-synonymous mutations, meaning that they are creating something that is totally different than what was being created before. And point mutations that don't alter the overall protein produced are called synonymous mutations, things that make the same thing as before, because sometimes, remember, I've got this amino acid wheel up here to remind you, but sometimes you can switch one of the nucleotides and you code for the same amino acid, so the same protein would be produced. So sometimes that happens, and perhaps more frequently than you might think. So point mutations are a single base change. Insertions and deletions, which we did talk a little bit about before, those involve the addition or removal of a single nucleotide, actually more than one nucleotide base sometimes. So insertions and deletions are classified kind of in the same group. So they're called indels for short. That's a smushing together of those two words. So indels involve the addition or removal of one or more nucleotide bases. If it's not a multiple of three, because remember, codons are three bases, ACT, GTT, whatever, those are codons. So if you were to insert three bases, it would not change all of the words that come afterward, all of the codons that come afterward. The codons themselves would remain the same. But if you insert just one or just two, or you delete just one or just two, then you start to change the reading frame. And to illustrate what a reading frame is and kind of the extent of how bad this can be, imagine that our genetic code that we're starting with is this sentence: the fat cat sat. I didn't come up with this, don't look at me. So the fat cat sat, that's our original sentence. Now what we have here is that in a later iteration, so right, we're trying to like copy, replicate, is that the T part of this sentence gets deleted. We accidentally lost it. Now, because codons will always be sets of three, now to make a full codon, And when we've lost the T, we've got to do HEF. We've got to take the F from the fat cat that was used to be part of the next codon. And so every single codon down the line changes. That is what I mean by a reading frame shift, and that sucks. That's normally a really bad mutation. Okay, so down here you can kind of just see visually in a less fun way, because the fat cat's hat is fun, right? But in a less fun way, you can see how this, let me get my pen again, this insertion has caused all of these amino acids down the line to be something different than what they were in the original form. And so this is really what we call this a frame shift. Frame shift mutation. A shift in the reading frame has caused all of these things to occur down the line. And so our protein, totally different, because now what has happened here is that we've got a premature stop codon. That stop codon should be way further down the line, but because all of our codons shifted, all of our reading frame shifted, we accidentally coded for an early stop, an early release of our amino acid chain. So the protein didn't even get fully formed. You can see the one that is produced after this frame shift mutation looks totally different and a lot smaller than this one that we started with. So frame shift mutations, which occur via indels that are not in multiples of 3. Again, if it's a deletion of 3 or a multiple of 3, you're only changing a singular section and not everything else down the line, because it'll read it as a codon that's in and of itself. So it'd be similar to me adding an additional word in this sentence. So the sentence as a whole would still make sense. I've just added an additional word, which could be confusing or could be helpful. Depends. So mutations can also occur with chromosomes, duplicating chromosomes. Trisomies and monosomies can occur during meiosis when things are not separated totally symmetrically like they should be. Meaning that the chromosomes don't separate into the daughter cells equally. So in an instance you might be familiar with is Down syndrome. Down syndrome occurs when someone inherits 3 copies of chromosome 21 instead of 2 of them. Sex trisomies are also something that occurs naturally in life. So while many of us were taught in school that sex, you are either XX or you are XY, this is a gross oversimplification of what biology actually looks like. Totally possible to be somebody who is XXX, somebody who is XXY, somebody who is XYY. All of those are survivable and fairly common. They can cause things like sterility or abnormal, what we would call abnormal sex development, but that doesn't mean that that person themselves is abnormal. It means that the trajectory of sex development is not what we typically see. That is what abnormal means. It doesn't mean there's anything wrong with it. It means that it's something different than what we typically see. Now, it is also possible that people can be just an X chromosome, singular X chromosome. That is called sex monosomy. So the prefix mono meaning 1, and trisomy, tri means 3, right? Like a tricycle. So the X chromosome does contain some genes necessary for life. So it is not possible for someone to be just Y, to just get a singular Y chromosome, because that embryo and fetus would not make it to be viable for birth, just because the X chromosome does have genes that are necessary for a living human being to function. But it is possible for someone to just have a singular X chromosome. Beyond this, and we'll get to it in a different lecture, there are a bunch of other genetic or hormone mutations that can cause someone to look more masculine or more feminine in their secondary sex characteristics and in their genitals. All of this variation in sex chromosomes is natural and normal, and it's all part of variation. Variation is a spectrum. It is rarely this or that. It is most of the time, it is this. It is a spectrum with infinite possibilities in between. All right, so the next force of evolution that we will cover is genetic drift. This is kind of a misnomer, because the things that are drifting is not the genes themselves, it is the alleles that are drifting. There's frequency of alleles that are drifting. So genetic drift is defined as random changes or drift in allele frequencies from one generation to the next. The random part is key here. I cannot emphasize this enough. This specifically, genetic drift specifically occurs when none of our types of alleles are gene variants. None of them confer a specific advantage or disadvantage in a particular context, right? And remember, evolution is only taking into consideration this context that you're currently in, this environment, this time, this climate. It doesn't think ahead down the line 20 generations. So an example I can give you is very, very early on in the primordial soup that organisms and life evolved in. There were amoeba-like cells that had two variants. There was one with a smooth cell wall shapes and a ruffled cell. So it looks kind of like, I don't know, it looks like a ruffle. It's got a skirt on, if you will. So it's got a bunch of these ins and outs and this ribbon-looking exterior to it. So the only thing that is different in these two particular cell types is the outside, the formation of the outside of the cell. Everything inside is the same. The components are the same, the cell functions the same. There is no difference in the ability to survive and reproduce between these cells, which means that there is not a selective advantage here. There's no pressure. There's no predation. There's no something favoring or disfavoring individuals reproducing and passing along either ruffled edges or smooth edges. So random inheritance patterns, random chance can still change the frequencies of these alleles, and that would be genetic drift. Now, there are two specific types of genetic drift that you should be aware of. The first one is a population bottleneck. This occurs when the number of individuals in a population drops super quickly, very dramatically, due to some sort of random event. Most familiar examples that you might be aware of are things like tsunamis, like natural disasters, tsunamis, hurricanes, volcanic eruptions. Think about Pompeii, right? So the eruption of Pompeii, Mount Vesuvius erupts. Now, people didn't really have an indication of this, and yeah, sure, some of them survived and some of them didn't. Like a lot of them didn't, right? But some people did get out. But there was no biological uh adaptation that allowed for that particular subsection of people who did survive to be better at surviving in that instance. It was pure random chance. Same thing if like there was a rift that opened in the middle of the earth and some people fell in, or maybe like a bunch of people, maybe like half the United States fell in and half didn't. That's random. It's random. It cuts down our population a lot, but it was a random thing, and there was no particular trait that determined who was better at surviving and who wasn't. So when a large portion of a population is wiped out, what we see is a huge decrease in genetic diversity, and that the allele frequencies in that tiny survivor population are often much different from the pre-disaster or what we would call the parent population. So again, to help you visualize this, I've got two different images on the screen. The first one pertains to our ruffled and smooth cells. So in the beginnings, we had quite a few more ruffled cells than we did, or smooth cells than we did ruffled cells. Now we had, in the second image conjoined to it, we had a volcanic eruption, and molten lava is wiping out a bunch of these cells. And you can see that who happened to not really be in the path of this lava? The ruffled cells. Most of them, or maybe all of them, survived, and a lot of the ruffled or the smooth cells did not. So by pure chance, not because of selective pressure, not because of natural selection, these guys, these ruffled cell guys, increased in frequency due to a random event. Now, down below that, I have an example that I tend to visualize in my head when I talk about this, and this is a bottle full of marbles. So our original or parent population in this glass bottle filled with marbles, heavily, from just looking at it, we can see there's a bunch of yellow and green marbles and not so many red marbles. Now, this visualization is awesome to me because you can actually see why we call it a bottleneck effect. And it's because this bottle's been turned over, and underneath it's a mason jar now, and that neck of the bottle, that's smaller than the body of the bottle, only lets some marbles come through. And so if we look at the marbles that fall through to create our surviving population after whatever bottleneck event occurred, now just by comparing, and it'll become more apparent here in a second when reproduction happens, but we can already see that there are certainly more red alleles or red individuals, red colored marbles, in this surviving population than there were to start with. So you give it a few generations of repopulation, and now what happens in our new population, our descendant population from those survivors, we've got a bunch more reds and very few yellows. So bottleneck events decrease genetic diversity a lot, which is bad for the longevity of any species. I've already covered this multiple times. But it also typically makes the descendant population of those survivors look quite different to what the population looked like before that bottlenecking event due to chance. A really famous bottleneck event that happened was the extinction of the dinosaurs, the Cretaceous Paleogene extinction, or I think it's called also the KPG extinction. And this occurred when an asteroid crashed into what is now known as the Gulf of Mexico or the Gulf of America, depending on who you listen to. Many species were immediately wiped out by the asteroid blast, but those that weren't had to deal with a lot of consequences to the environment. So the skies just got filled with dust, debris. The temperatures just started to plummet because a lot of the sun's rays, which are what heat up the land, were blocked out by just like this massive dust cloud. And about 75%, that's insane, 75% around of the world's species went extinct as a result of the impact. But on one level, we have to be thankful for this because this is what started the era of mammals, and we are mammals. So if this didn't happen, who knows if humans would be are so prolific a species as we are now? Because at the time, mammals were largely like these little tiny creatures that lived underground. We couldn't compete. Our ancestors couldn't compete with the dinosaurs. So we were tiny little rodent creatures and we lived underground. But so when all the sun's rays get blocked and everything gets dark, who is used to that? Who can hide from inhaling that? Who's used to the darkness and the cold? The mammals that have been living in caves and underground. So as the creatures that were who needed the sun's rays even more than these underground mammals did, as they died off, it opened up, and the environment started to stabilize. It opened up a whole new niche for mammals to be able to come out and be like, oh my gosh, all this whole world that I couldn't have seen when the dinosaurs were around because I was going to get eaten or stomped on, now I can. And so mammals start to take over the really many of the places of the globe after this. And down the line, many millions of years down the line, enter us humans. So history could have been a lot different if this didn't happen. But definitely a huge and very famous population bottleneck. We can also accidentally cause population bottlenecks to occur. We cause them to happen in bacteria populations. We have created, on accident, antibiotic-resistant bacteria. So what happens is when you get a bacterial infection, if you get I think there are different kinds of bronchitis, but definitely one of them is bacterial. Then you will get prescribed antibiotics. And most of the time, your doctor is going to give you a regimen that is like 10 days or something over a week. And they're going to specifically tell you, finish this. You're going to feel better, but finish these pills anyway. And I've actually heard of people like when they feel better, they save the rest of the pills so they don't have to go to the doctor later. That is so bad. Please do not ever do that. I understand that the medical care system in this country is horrible and it's expensive and it shouldn't be, and I get that, but you are setting yourself up for horrible things to happen in the long term because you need that whole regimen. The problem is when you start to feel better, you might forget or you don't think you need the pills anymore, so you stop taking them because some people don't want to take a bunch of pharmaceuticals. That's totally fair as well. That's a fair, valid viewpoint. We are over-medicated in this country. But the reason why this is a big mistake is because antibiotics quickly kill off a pretty big percentage of the bacteria, which is why your symptoms get better pretty quickly. But this has created a population bottleneck, in which a small amount of that bacteria usually survives the early days. If you take your full regimen, odds are it might be strong enough to wipe out the rest of them and prevent that resistant strain from proliferating. But if you don't, odds are that resistant strain is going to become more and more and more number. They're reproducing quite quickly. And now the next time that you get sick, now the next time that you've got enough of them in your body that you start to feel crappy and you go to the doctor, they're going to give you the same antibiotics as before. And guess what? This time it's not going to work. So now they've got to try a new drug on you. Now the problem is that sometimes this happens to people a number of times, and now they've got a strain of bacteria inside of them that is resistant to multiple medicines, to every medicine we have available. And this is where people die. And a lot of these antibiotic resistant strains, these exist in hospitals just because they are such sterile environments. So by over sterilizing our environments, we are in a co-evolutionary battle. We try to get rid of all the bacteria, and then they evolve a mutation because they're reproducing way quicker than we can get medicines on the market. So they evolve a mutation, and now our medicine no longer works. Then we get a new one out there, we dwindle them down, but now there's a resistant strain. And it's just a co-evolutionary thing. So while, I'm not going to condone I'm not going to say go eat things off the ground. I'm going to say be cognizant of how often you're putting hand sanitizer on your hands. Because if you're using it religiously many, many times a day, you might cause yourself to get one of these resistant strains that's going to be very, very hard to eradicate down the line. So modern medicine is great, and these things that we've got to keep environments and kids from getting sick are great. But also, if you don't expose yourself to the less harmful bacteria, the lesser strains, you don't build up immunity to be able to take out the greater strains inside your body. So grain of salt, right? There are many things. It's not just the antibiotics. There are many things that cause this type of bottlenecking to occur, and then the new population looks way different than the original bacterial starting population. So things like overuse of hand sanitizer, sterilizing products, inappropriate use of antibiotics as preventative measure in livestock. So sometimes people just give all their livestock antibiotics just to prevent them from getting sick so that they can slaughter them and sell their meat and stuff. But then they get the resistant strains, and sometimes because viruses and bacteria mutate really fast, sometimes those strains can hybridize, and then they get into humans, and we have an epidemic, right? Then we have like a COVID happening. Also, if you go to the doctor and you say, give me antibiotics, and they're like, I can't, it's a viral infection, I can't give you antibiotics for that. You listen to the doctor and you say, okay, you know what's best, doctor, because antibiotics don't work on viral infections, and you should not be taking antibiotics unless you actually need them, because you can cause what I'm talking about to occur. The other kind of genetic drift that I want to talk to you about is founder effects. So founder effect is the singular, but I'm kind of talking about the effects in general. So I've put an S here. But founder effects occur when members of a population leave the main or parent group and form a new population by going somewhere else that no longer interbreeds with the original population. So like in population bottlenecks, that new founding population oftentimes, just by chance, has different allele frequencies than what you saw in your original parent population. So alleles that were once rare in the parent population can become very prominent in the descendant population of those founders just by pure chance, because those are the ones that happen to go somewhere else and reproduce. Even recessive alleles can become quite proliferative in these smaller groups. So we can see we had an ancestral population of butterflies that was fairly spread, right? Like evenly between white butterflies, orange butterflies, and brown butterflies. But this group of colonizers, that's what they're called, sorry. But this group of colonizers to this other island, they say, you know what? There's too much competition where we're at for nectar and access to flowers. I see landmass right there. I'm going to go try to my luck over there. So they go colonize this new space looking for a better life and better access to resources for themselves. But now this founder population doesn't have any white butterflies. They've got 2 brown butterflies and an orange butterfly, which means in all likelihood that my descendant populations of that founder group may have little to no white butterflies at all. So it's going to look totally different than my original parent population. So founder effect is really closely linked to the concept of inbreeding. And by inbreeding, I don't just mean reproducing with someone who's your family, who's related to you. I know that's how this word is typically used, but in our sense in this field and in genetics, it means something a little bit different. It means mating that's happening in a group that is a very small, closed population, meaning that they are breeding within this tiny group. There once was a big group, and now they're breeding within this little tiny group. And so it means that the same traits are coming, I call them marbles here, but like the same marbles are being picked out of a bag again and again and again. And it's without variation, without genetic diversity, without new traits coming into the population. If there's a recessive allele in here, or multiple recessive alleles just by chance, you can cause serious like deformities and could eventually like the population could dwindle down to nothing due to it. So having genetic diversity, as I have emphasized multiple times in this lecture and will continue to emphasize in general, is important to the longevity of any group. So a group with limited allelic variability, a group that is not very diverse, is going to have some genetic issues down the line. So a small population means that we're taking those same marbles, which might be mutations or like bad deleterious mutations, and we're pulling the same ones out of a bag again and again. And we can't change that because we're not getting any new alleles coming into our population. So we are losing genetic variability. And then what happens is we start to lose fitness because these mutations are starting to add up, and now we're just getting the same ones, but all in multiple individuals, the exact same mutations, and maybe they all become infertile. And in general, any like inbreeding instance that goes on for a very long term tends to lead to lower reproduction, higher mortality, and more deleterious mutations in general. This doesn't always happen, but typically this is the trend that we see. So we can see this in small, physically isolated populations, but it can also happen when cultural practices limit mating to a particular small group. So inbreeding increases the risk of inheriting two copies of any non-functional or mutant allele, and that can cause problems. I will give you an example. So an example that you may or may not be familiar with are the Amish in the United States, who they have a very unique history, they have unique cultural practices, and they emerged from a very small founding population. But over time, since they've been here really in the United States, they have tended to select mates from within their group. And people tend to do this in general, right? Like you want somebody who understands your culture, understands your religion. It makes getting along with them and liking them easier and relating to them. So this isn't anything different than anybody else would do. But so the population in the United States that I'm talking about, they are an old order Amish group and they currently live in Lancaster County, Pennsylvania, and they have approximately around 50,000 members today. But all of those 50,000 members can trace their ancestry back to about 80 individuals. So this was a very small founding population, right? Fewer than 100 people. And they immigrated to the United States from Switzerland in the mid-1700s to escape religious persecution. They heard that they could openly practice their religion here, and they said, we want to do that. So the Amish almost exclusively select mates from their own communities, and we see the effects of this in the fact that they have very high numbers of recessive traits that are then compared to the overall population of the United States. One of the most common genetic conditions observed in the Lancaster County Amish is something called Ellis-Van Creveld syndrome. And this is a recessive condition, but it's very prevalent because a lot of these individuals are carriers, which means that your odds of inheriting two recessive alleles go up astronomically. And then if the more and more homozygous recessive individuals you have, the more certainty that more and more offspring will carry this condition or express this condition. So this Ellis-Van Creveld syndrome is characterized by short stature or dwarfism, polydactyly. This is another good one where your Latin terminology can come into play here. Poly means many, like polygon or polyamorous. It means many. And dactyl just means like digits. So it means many digits or more than 5 digits. Abnormal tooth development like enamel issues and heart defects. So among the general world population, this syndrome is seen in about 1 in 60,000 births. And among old order Amish in Pennsylvania, we see this in about 1 in about 200 births. So the comparison then, 1 in 60,000 to 1 in 200, clearly the effects are magnified in this population. And this is an example of how genetic diversity, the lack of it, the lack of incoming new genes, how that increases your risk of genetic diseases, which means that genetic diversity, having a lot of different alleles in a population, decreases the risk of genetic disease. Moving now to gene flow, our third force of evolution. Again, this is a misnomer because alleles are the things flowing, not genes. But gene flow refers to the movement of alleles from one population to another. In most cases, this can be synonymous with migration when we're talking to people, but really it means that there are two groups that perhaps were not interbreeding before, and now they've come into contact with one another, and now they are sharing alleles. So genes are now flowing where they weren't before. So I've got my ruffled and my smooth cells in this particular image on the screen. Our lava from our, what was that, population bottleneck, has now cooled, and now grasses are growing on it, and now the cells that were once separated apart from one another can now re-emerge and interbreed with one another. And so you can see these smooth cells that were stuck on the opposite side, isolated from everybody, can now come around and say howdy do to the rest of the cells. And so they were separated, their gene pools were separated by a physical obstruction. Now that physical obstruction is gone, and those two populations can now share alleles again. They can create a bigger gene pool, and now technically alleles are flowing, but gene flow is occurring. So in figure 1, there is no gene flow occurring because they are two separate populations geographically, even though they're the same species. But in figure 2, we do have gene flow occurring because that obstacle is now traversable. So among humans, gene flow gets its own name, and that is admixture. We are all members of the same fully breeding compatible species. We are so genetically similar. In fact, I've mentioned it, but we are the same even subspecies. We are Homo sapiens sapiens. So Homo sapiens is the species, sapiens is the subspecies, the third word is the subspecies. And in the past, we all used to be isolated from one another on different continents, right? But as we started migrating, different groups came into contact with one another, and we can track these admixture events in history. And there are still some isolated populations today, but much fewer than in the past. And when or if they do admix with other groups, we would be able to see that in changes in allele frequencies as well. Moving finally to natural selection. Natural selection we have talked a little bit about, but I'm going to go into more depth about different kinds of natural selection. But this occurs when certain phenotypes confer an advantage or disadvantage in survival or reproductive success. Alleles associated with those phenotypes will change in frequency over time due to specific selective pressures. Whether a trait is advantageous or whether it's harmful is not something that is fixed in time. It depends on what environment you're in a current geotemporal context. So an allele can be helpful and become super prevalent in one particular context, but then if something changes, like a new predator arrives or something like that, it could become harmful. So there is no fixity here. This is why anthropologists love the idea of dynamism. You have got to change with the changing context. So in this image that I have on the screen, we are looking at an example of a selective pressure. We've got a crow who says, yum, green beetles are favorite. But so they're eating the green beetles. So as generations go by, we've got more and more orange beetles because they're surviving. Eventually, though, they're going to have to eat the orange beetles. But within these 3 generations that we see on the screen, green beetles have decreased because the crows were preferential to them. There is predation. That's the selective pressure. There is predation that is making the orange phenotype the more beneficial one. So that starts to proliferate over time. So natural selection, key here, natural selection operates on phenotypes, not the alleles themselves. It doesn't handpick those dominant or those recessive alleles. It only works and operates and recognizes the phenotype, the actual expression of whatever genotype you have. So there can be disadvantageous or deleterious alleles that are retained by heterozygotes because there's no negative effect. They're being masked by the dominant allele, so they just ride along together. So and we'll cover an instance where that occurs, but it's because the heterozygous condition is favorable. So we will cover an instance where the heterozygous condition is favorable, but just so you know, like recessive alleles that are horrible for you, that they can just be hanging around because the dominant one is typically masking them. So there's no selective pressure, really like strong selective pressure to get rid of it, except in the homozygous recessive individuals. All right, I warned you in, I think, the last lecture, but I warned you I'd come back to the peppered moth because this is the quintessential example of natural selection. So let me set the stage where we are in the story. We are in England during the Industrial Revolution, so in the 1800s. And before the Industrial Revolution occurred, we've got a peppered moth population. They are called that because of their coloring. It's speckles of light and dark. And they camouflaged really well on these like birch trees. You can see it on the image on the screen, or maybe you can't because he's hiding so well, but he really blends in with like the white and the gray of the tree. So that's the peppered moth version. He does great at hiding. So when birds were flying around looking for food, they couldn't really easily spot these moths and eat them. So camouflage is good pre-industrial revolution because the trees and the moth are both light and with speckles of gray. All right, but they weren't the only phenotype that existed. There were also dark color moths. Dark color moths had more melanin or pigmentation in their wings and in their coloring. And so these black moths were a lot easier for the birds to spot. You're doing a quick flyby of these trees. You see something stand out as a dark spot. You go to try to eat it because you think it's probably a bug. So these black phenotypes of the moths were being predated upon at high frequencies. So what happened to this type of expression, this phenotype? It starts to decrease. If all the birds are eating them, then they're not going to survive to pass along their genes, and they will decrease in frequency. So this particular phenotype before the Industrial Revolution had a very large disadvantage. Now, enter the Industrial Revolution. We've got huge factories. Nobody understands that environmental pollution and degradation is going to be a long-term problem yet. So there's no regulations, there's no EPA, there's no nothing like that. We're just spewing smog, pollutants into the air. We do this enough and it starts to coat things in our natural environment. It blanketed the countryside, made it like a dull and a gray. Think about ash from a fire. It was settling on trees, it was settling on leaves. And so what happens is that trees that used to be fairly light in color are now quite dark, like soot-like in color. And now what happened? Now the black moths can hide really well in the trees. They blend in, and the peppered moths, the lighter color ones with little speckles, stood out like lights on this tree. So birds always are like, I want the easiest thing to eat. So now instead of going for the black moths, they are now eating predominantly the peppered moths, the lighter color moths. So now we see a switch is that the peppered phenotypic variation becomes disadvantageous. It starts to decrease. The dark allele becomes very advantageous and starts to increase. So we get a switching of what is beneficial and what is not as far as phenotype. So by 1895, by the turn of the century here, the black phenotype accounted for 98% of observed moths. That's how well it served them. 98% of observed moths were this black phenotype. But is that the end of the story? No, it's not. So in the 1960s, finally, the world is catching on. We need to take care of our environment or there is going to be some big problems coming up. So there are environmental regulations put into place. Air pollution starts to decrease, so it starts to get blown off trees. We're not laying down new layers. So now the trees start to slowly return to their former lighter color. So how do you think the peppered moth population responded? Well, in 2003, the maximum frequency of the dark phenotype was 50%. Remember, the midpoint here during the Industrial Revolution, we were at 98% black phenotype. By 2003, that's about, 40 something, 43 years later, we've got down to 50%. And then later, we've got down to 10%. Well, in some places currently, down to 10%. So you can see this is how I was explaining that certain traits that are advantageous versus disadvantageous, there's no one fixity. One is advantageous right now, then later becomes bad, then it becomes advantageous again. And now, so peppered moths are now favored if the trees look like this. However, that's general natural selection. There are a few specific types of natural selection I have to cover. So those are directional, balancing or stabilizing. Those are the same. Those refer to the same thing. They're synonymous. Disruptive or diversifying, and then sexual selection. So balancing or stabilizing selection occurs when selection works against the extremes of a trait and it favors the intermediate, the middle of the road version. So a great example of this is birth weight in humans. So there is a need for babies to be small enough to fit through the birth canal and not cause issues to the mom, right? Because then if mom dies, then her fitness is gone. And I'm not talking as a human. Obviously, that's very sad when there's maternal mortality. I'm speaking from an evolutionary perspective. That evolution doesn't really care about the human in an emotional way. It just says fitness. Fitness is the goal here. If you die of big-headed baby, fitness goes to zero. We don't want that. So, but at the same time, we need babies to be big enough to maintain a safe body temperature after they're born, for their brain to be, and for their brain to be developed enough that they can maintain their bodily functions, such as body temperature. So what we go towards is like what I call like a Goldie, I'm sure other people do too, like a Goldilocks perspective. We want the middle of the road, not too big, not too small. Um, because if the babies are too big, right, then we have birth complications. I've got another example on the screen that might help you more. This is an example of a robin laying eggs. So if robins lay too few eggs, then something might happen where none of those chicks survive. So they want to have bigger clutches to maximize the number of chicks that might survive. But if they have too many eggs, like if they're one pair of robins, actually, I don't know if robins raise their young together or if it's just one of them, but I'm just going to assume it's both. If they have 20 eggs and they're just two robins, they can't take care of all of those chicks to the same capacity. So some of them are probably going to be malnourished, not able to survive, sickly. So what evolution favors and what natural selection favors is the middle of the road, the Goldilocks number that's not too big and not too small. So it maximizes the number of chicks that parents can successfully rear so that they can then go on and reproduce. To give you another human example, where the heterozygous condition is favored, but the recessive allele is not a great one, I'll talk about sickle cell anemia. This is one of my favorite examples to discuss because it's unique. And it offers you like a way to work through the problem logically. Sickle cell anemia is a genetic disorder where if you get a copy of an allele for sickle cell inherited from both of your parents, every single red blood cell in your body is sickle shaped. This means a couple of things. This has a couple of consequences here. One, that your cells can't carry oxygen as well as they could as if they were this normal round blood cell shape. And also, they are, because they're sickled, it's like monkeys in a barrel. They get caught on one another. These round cells can bump into each other, go over them. But if they're sickled, they catch on to one another. And what can happen is that they are very likely to cause blood clots, which can be painful and sometimes very dangerous. So a great question to ask to work through logically is from an evolutionary perspective, understanding that actually many people who have sickle cell anemia don't live past their 20s just because they throw a lethal clot. If this disease can be often fatal before that person has reached their reproductive potential, why has natural selection not eliminated it or at least made it extremely rare? And there is a reason for this, and this has to do with the heterogeneous genotype. So if you have, two alleles for normal red blood cell, this is what they'll look like. This is if you have two alleles for sickle cells. And then the heterozygous, you get a blend. You get a 50-50 blend of these type of red blood cells and these type of red blood cells. And the question now becomes, why might that be beneficial? Because I told you that the heterozygous genotype is what's going to be selected for. The pressure is going to be for the middle of the road phenotype. But why? Okay, so we ask some sub-questions. We use the scientific method here and we ask some sub-questions. One, where does sickle cell have the highest incident rate? Where do we see most of the people who express sickle cell, whether they're carriers or whether they have sickle cell anemia, which is when you have two copies of the sickle cell trait? Well, we look and we see there are very high concentrations of this trait, this phenotype, and these traits, these allele frequencies, in certain parts of Africa. Now, what else seems to be fairly prevalent in those regions? And now, if you were a scientist, it'd probably take you a lot more guesses before you get here. But the guess that turns out to be correct is the correct guess is that malaria, if you put the map of malaria over this, you tend to see some really large areas of overlap. So malaria is a mosquito-borne illness that can also be quite deadly to humans. People with one copy of the sickle cell trait have a strong resistance to malaria, and this is adaptive. And as a byproduct, because having one allele for regular red blood cells and one allele for the sickle shape is helpful to surviving malaria, this sickle cell anemia, when you have two copies of the sickle cell trait, that just hangs around as a byproduct, as an unfortunate kind of consequence of selecting heavily for the heterozygous condition. So here we've got the life cycle of the mosquito, and basically it uses the red blood cell as a reproductive mechanism. It reproduces within those red blood cells, but in the sickled red blood cells, it can't do that. It can't reproduce inside of there. So people who have one sickle cell trait, they normally don't get super sick from malaria or die. So it prevents death via malaria. So why is this important? Well, if you try to eradicate sickle cell anemia, if you're like, I really think it's horrible that people have sickle cell anemia, like I want to find a way to try and maybe cut the genome like that's called CRISPR, people trying to cut particular parts of the gene out and put it like a healthy gene in. Well, if you try to do that, then you decrease malarial resistance, which apparently is very necessary in this region, or natural selection wouldn't be exerting so much pressure to select for the heterozygous condition. So this is where the idea of holism comes in, is that everything is connected. You can't just say, oh, simple solution, I'm going to figure out a way to get rid of the sickle cell allele or the coding for sickle cell. You can't. You can't without also thinking about the consequences for malaria and people dying of malaria. So you have to look at a problem from multiple perspectives. So why then does this study represent a case of stabilizing selection? Well, remember that stabilizing selection selects against extremes. In this case, that would be the whole red blood cell and the double sickle cell, and it selects for the middle, the heterozygous condition. So if you're homozygous for sickle cell, you probably have anemia. You're probably going to throw a clot and have likely an early death, which means from an evolutionary perspective, you have low fitness. But if you're homozygous for red blood cells, for normal red blood cells, then you probably are susceptible, you are highly susceptible to malaria. You likely will have an early death if malaria is acquired. So that's not great for fitness either. But if you're heterozygous, then you have some sickle cell, which means it's not fatal to you. You're probably going to survive. You'll be fine. And you have some normal cells, and having some sickled cells confers you some selective advantage against malaria, from dying from malaria. So the middle condition, the heterozygous condition, is the most beneficial. So it's selected for. The next kind of natural selection is the opposite. So the heterozygous or middle of the road condition is selected against, and the extremes are selected for. So this is our original population, is like a bell curve, a typical bell curve. But now our descendant population, after there is natural selection against the intermediate condition, it now looks like two humps, like a two-humped camel, right? That middle peak of the bell has gone down where it used to be the peak, and now it's it is the minimum. So sometimes this goes on for long enough that it can lead a population that simply has different phenotypes to actually become different species. So this is pretty different. Here in this image, we are looking at different populations of rabbits, and they are hiding in rocky environments. And in this rocky gray environment. This super white rabbit is very easy to spot, but it's the heterozygous condition, whereas these other two are not. Those are the homozygous conditions. And so the heterozygous, which is the very, very white rabbit that's easy to spot in the Rocky Mountains, when there's not snow, then it gets selected against. This type of speciation that I'm discussing, how it can lead to speciation, is kind of what Darwin was thinking about when he was discussing the finches in the Galapagos. He thought that his finches, now what we call his finches, I don't think he called them his finches at the time, but he thought this is what happened, that there was diversifying selection and that it eventually led to speciation, because he saw that, I mean, there were multiple species of finch that were eating large seeds that all had big beaks to be able to crunch through those large seeds. And then he saw there were multiple species of finches eating small seeds, and they had small beaks because they don't need to develop these huge beaks to eat these tiny seeds. But he noticed that there weren't really any beaks that were like medium sized, the in between. So they were really large and really small, but there was like no intermediate. And then he noticed that there aren't really any medium-sized seeds. They're like either giant or they're tiny. So birds who had medium-sized beaks probably couldn't survive. They couldn't eat the big seeds. Their beaks weren't big enough. They weren't dexterous enough to be able, like, think about tweezers. If you had like oven mitts, it'd be hard to pick up tiny seeds, but if you have tweezers, it's easier. So they probably couldn't eat the small seeds. They couldn't eat the big seeds. There was no intermediate seed for them to eat, so they probably died off. So there was selection against the intermediate phenotype. So over time, he thought these two groups, my two sections of my curve, my two camel humps, right, that they became so different that they became different species. Next, we have directional selection, where environmental pressures favor one phenotype specifically over a different phenotype or over any other phenotype. And it can cause the frequencies of advantageous alleles to gradually increase. So in the case of the peppered moth, the direction shifts multiple times here, like the black phenotype is helpful, then the peppered one is helpful. So the phenotype shifts, but there's clear pressure towards one or the other. So we've got an initial selection for lighter pigment. Then we've got pollution happening. We've got a selection for darker pigment. Then we've got environmental regulations, and we've got selective pressure for lighter pigment again. Finally, we've got sexual selection. This is an aspect of natural selection where selective pressures specifically affect reproductive success rather than survival. And success here is the ability to breed and raise those offspring to be of breeding age. So sexual selection favors traits that will attract a mate or secure a mate for an organism. And sometimes these sexually appealing traits can carry really big risks in terms of survival, but you got to risk it for the biscuit sometimes. So we can look at birds first. What pattern do we notice here with regard to male birds? So the male is on the left in each of these photos. We've got a bright blue and white bird sitting next to a brown and white bird on the right. Then in the middle, we've got some ducks. We've got a male duck on the left that is bright green and brown, and the one on the right is a female who is muted brown and a little bit of white. And then on the very right, we have a peacock and a peahen. The peacock has a huge tail, and the peahen has a very small tail with not so much color in it. So in this pattern, you can see that males tend to be the brighter of the species, and this does largely hold throughout all of the bird world. So I'm going to narrow in here on the peacock peahen example just to walk you through this, but and maybe you didn't know that females are called peahens. I wasn't sure how to phrase that, but females are called peahens and males are called peacocks. So in mating season, males will fan this beautiful, huge, colorful tail in front of the peahens, and they'll strut in front of them like, look at all these feathers. Aren't these great? The peahens will selectively mate with the males that they find the most appealing, which tend to be the males with the biggest, most wonderful, giant tails. Well, how might you explain this? What might these big, colorful tails indicate to peahens? They probably don't like the color just for the color. There's probably something else going on here. What might full, colorful tails indicate to peahens? Well, one thing it might confer is health, because animals are not able to make beautiful colors if they do not have enough nutrients to do so. This is the same in colorful fish too, and in apes, and in most animals. Your body will not make these big, bright, beautiful colors unless you are healthy enough to do so, unless you're meeting your dietary needs. So if you're sick or you're in starvation status, your colors will be more muted. And I don't mean you as a human, although that might be true, but I mean in general in animal species. So for females, it might indicate something about health. They are healthy, they're not sick, they're able to find enough food. Now, so we know that females select for big, beautiful, giant tails. So these confer a reproductive advantage, but there is a burden because if a male gets if a predator locks onto a male peacock, that's redundant, peacocks are male, but it locks onto a peacock and he is trying to catch him, that huge tail, it weighs something. And trying to escape a predator with that tail is very difficult. So, some people argue, many scientists argue that this is part of the allure for the peahen, is that that means that that male has been able to carry around that big, beautiful tail, that weighty tail behind him, and he hasn't gotten eaten yet. So that means he's pretty good at surviving. And peahens want their offspring to be good at surviving. So if she selects a male that has this big, beautiful tail, which means he's not sick, he's healthy, and he's been able to escape predation, they are giving their offspring the best chance at survival. So she selects to mate with the male who impresses her the most with the tail. However, it is important to remember that sexual selection relies on the trait being present in the mating years. If a trait is not present there, then natural selection doesn't need to operate on it. So an example of this in humans is neurofibromatosis, which is a genetic condition that causes the growth of a lot of benign, meaning non-cancerous tumors called neurofibromas. This is actually more common than you might think. It's diagnosed in about 1 in 3,000 babies generally. But this condition can be extremely disfiguring. I've got an example of two individuals on the screen, meaning that their bodies, and sometimes just the faces, depending on the variant, or their entire body can be covered in these different kinds of benign growths. So the problem is, people might ask, like, if this person knows that they have this condition, why would they willingly have genetic offspring? Why would they have their own biological offspring? Like, why would they want their offspring to have the same condition that they did? Like, doesn't that seem kind of selfish? Well, the answer to that is that neurofibromas don't all appear at once. They grow slowly and they only start to appear during puberty. They are few in number. They are kind of slow to grow. The full extent of this condition is not experienced until people have already started families. So for many, they might not be diagnosed or know that they have this condition or that is genetic until they have already reproduced. So at that point, no takebacks, right? It's already been passed down to the next generation. So natural selection can't act upon this because it's already been passed down. It doesn't occur, it doesn't become aware or manifest until after most people's reproductive years have already passed them by. So finally, we've hit all these aspects of natural selection. I want to give you, kind of zoom out and give you like a scale and kind of put this into some other terms. So we've been talking about allele frequencies. That's where we kind of started at the beginning. We talked about allele frequencies. That level of evolution is microevolution. That is changes in allele frequencies in breeding populations on a relatively smaller time scale. But if you zoom out and you look at generations and generations and generations over millennia, now you're looking at macroevolution, which are these changes that accumulate and end up into different species, into speciation events, basically. That, you can think of Darwin's finches, their selection against middle-sized beak, same species so far, but we get strong preference for small beaks, strong preference for large beaks, and eventually they become so different they can no longer mate. That's speciation. So we can return to the peppered moth study to kind of put this into context. So what if environmental regulations clean up part of the environment, but not all of it? So one pigment's favored in one location and the black pigment is favored in a different location. Well, this could lead to speciation. If those two populations are geographically distinct from one another, they don't live right next to one another, they live on different trees, they're not talking, they're not interbreeding, then eventually they can accumulate tiny mutations over time. And they get enough of them that they can no longer interbreed and they become two separate species. So speciation level events are macroevolution, and microevolution is those smaller changes over time in allele frequencies. So just to reiterate, speciation, okay, I'm going to give you also two examples of specific kinds of speciation events, but speciation occurs when a single population divides into two separate species. So the two types of speciation we'll cover are allopatric and sympatric, and the prefixes here are very important. So allo means different or distinct. So in primates, there's a form of parental care that's called alloparenting, and actually that might be something that exists in other groups as well, but I'm most familiar with it in the human and other ape context. Alloparenting, it means that individuals besides the genetic parents are helping to take care of offspring. So if you were ever like for the beginning of my life, my mom raised me with my grandparents. My biological dad wasn't in the picture. So my grandparents weren't my biological parents, so they were allo parenting me. They were still parenting me, but they weren't my genetic parents. So they were distinct from my genetic parents, so they were allo, they were different, but they were still taking on that parenting role. So allo parenting. Anyway, I'll reinforce that when we come back to it, but allo means different. So allopatric speciation is caused by long-term isolation or physical separation of subgroups of a population. So something in the environment might occur that prevents individuals from they were breeding and now they can't breed with one another. So maybe a river changes course, or an earthquake causes a rift, or a lava flow occurs, or something like that. And over many generations, mutations and adaptations give those two subpopulations they add up enough that they are so distinct they can't produce fertile offspring, even if that barrier is one day removed. I've got that illustrated on the screen here. So the illustration is showing shapes. In image A, we've got a single population on like an island landmass. They are all circles. They are all the same species. That's what the circles indicate. Something happens and some tectonic plate shifting happens, and these two groups split into two separate groups. But they're still the same species. Not enough time has passed. If these two land masses bumped into one another, if they're birds and one bird flew to the other landmass, they could still interbreed. However, now as more time is passing, they are accumulating mutations separately. They are not interbreeding, so they start to change on their own. And then if one day those two land masses come back together, they are now so distinct. One is a triangle now, and one is a filled-in circle. They can no longer share, they don't have the same amount of chromosomes, or maybe they don't have the same mating tactics or something like that. They can no longer reproduce. We have a speciation event. But the allo part is that they were kept separate from one another. The separating, the distinct geographic locations between one another, that is what caused the speciation to occur. So sympatric is the opposite. It happens when species are in the same location. There is no physical barrier that's keeping them from reproducing with one another. So this typically results from a new mutation that pops up among some members of the population and not others. And it prevents them from reproducing with anybody without the same mutation. And this is fairly common in plants. It can happen where they create, the mutation is that they just have a different number of chromosomes. And it happens so frequently to the point that those plants with a different number of chromosomes still find other plants that have the same number of chromosomes because they've accidentally got the same mutation, and they can still reproduce with one another. But it could also occur in like birds. If a mutation occurs in a couple of birds where they can only hear like maybe higher frequencies, and so now those birds can only mate with one another because they can't hear the lower frequency mating sounds of a different bird, it can happen that way too. So there are different kinds of mutations that could be behavioral or genetic that cause only certain subgroups of the population to be able to continue mating with one another. So even though they're all these birds are living in the same forest, these ones can only hear the mating songs of these other ones, and this one can only hear the mating songs of the original unmutated group. And so they can't mate with one another, not because they physically can't at this point, but because they can't hear each other. They can't understand each other. And over time, the differences again accumulate and they become two separate species. That is sympatric speciation that occurs in the same location without a physical barrier preventing interbreeding from happening. Finally, the fastest kind of speciation is adaptive radiation. Subgroups of a single species quickly diversify to fill a vast variety of ecological niches. That is what adaptive radiation is. Niche is what we use for the environmental ecological situation, and that's a set of constraints and resources that are available in an environmental setting. So imagine we've got a founder effect, right? And we've got a group of birds. And then some of those birds, so imagine that's like a bajillion more of these, right? A bunch of dots. And then a group of these birds go to an uninhabited island. They're like, oh my gosh, we're going to try our luck at this island over here. Well, they go to this island and they realize, hey, there's different things we can exploit here. Some of us can eat insects. Some of us can eat seeds. Some of us can eat sap from the trees. Some of us can eat small rodents. I don't know if a bird can do that, but I'm just imagining. So they start to because they don't want to compete with one another. Species generally do not want to compete with one another unless they have to because it's energetically expensive. We all want to be as lazy as possible, and I mean that from an evolutionary perspective. We don't want to waste calories unless we have to. So they start to say, hey, okay, well, I like bugs the most, so I'm going to go eat those bugs, and they start to develop adaptations to that. Now, a couple other species are like, well, I'm gonna go eat, seeds. Then they start to develop mutations to that. And then these ones are like, okay, well, I'm gonna go eat sap. And they start to develop mutations for that. So what it looks like in in an evolutionary perspective, is we've got a singular species that comes to a place that has a bunch of niches available for them to quickly fill. And then we see a very rapid speciation event as a bunch of different species come from this original singular species in a shorter amount of time just because there simply are so many niches available. If you were looking at like a evolutionary tree diagram, that is what an adaptive radiation event would look like. That is what an adaptive radiation event would look like. A singular species quickly kind of fireworking off into many different species because they are all evolving so quickly to meet the new environments that they live in. So they want to be in different niches so that they don't have to compete with one another. So to return to our Galapagos Islands, we are not sure how the first finches arrived there, but when they did, they found a bunch of things: insects, tiny seeds, fruit, varieties of cactus. And so some members of the initial population carried alleles that gave them distinct advantages to eat one or more of those things. And they were thus, because they all kind of congregated, all the finches that could eat cactus really easily, cactus flowers, they all started hanging out at the same place. So they were more likely to mate with finches that shared the same dietary niche. They're all exploiting the cactus together. So these differences, as they start to self-isolate and start to mate with only the other species that are hanging around the same places, they snowball, and differences between subgroups get bigger, and eventually we've got what, in the scheme of things, should take a very, very, very long time, now takes, a fraction of the time, and we've got rapid speciation. All right, that's it for today. Thank you so much for hanging around. I hope if you have any questions, let me know.